And we'll start with the pyruvate dehydrogenase complex. Again, this is in the mitochondrial matrix. Very inside of the mitochondria. All right, so there are 3 different reactions that take place within that complex. The first one, which is known as E1, or it's also known, and this is where it gets a little bit confusing, it's also known as pyruvate dehydrogenase. So it's the pyruvate dehydrogenase within the pyruvate dehydrogenase complex. Okay, it contains, again, there's a lot of cofactors in this. It contains thiamine pyrophosphate. So that's another vitamin. I think we talked about it, I think, when we talked about enzyme cofactors a little bit. That was the one that the disease is what, beriberi, and it was discovered by Nicholas Kulp, who was a subject of a Rembrandt painting. I think I went there. Okay, so what is this thing? The business end of this is all right, so that's the thiamine pyrophosphate molecule. I guess I drew it all for the sake of completeness. But the business end, so this is a plus right here, is this thing here, which is known as a thiazolium ring.
That's the circled one?
That's the circled one, yeah. What makes this kind of cool, you know, very, very heterocyclic ring here, is that this hydrogen is relatively acidic. Generally, you think of a hydrogen coming off of a carbon, it's not very acidic, right? You know, with pKa is like 35 or 40 or something like that. You know, it's not very acidic. But this one is actually acidic, and you can generate a carbanion right here. You know, so a carbanion, generating a carbanion under physiological conditions, that's not a trivial thing to do. So that's why, you know, we basically bring this molecule in to provide that chemistry. So what happens is, you know, how does this actually work? Well, I'm going to go ahead and, I'm not going to draw the whole thing, but I'm going to go ahead and draw the anion. So there's that anion, and it is going to attack this is pyruvate, kind of drawn upside down. So it can basically attack there. This becomes a leaving group. So we're going to pull carbon dioxide off. And now it is covalently attached here. And this is all, this thiamine pyrophosphate is tightly bound to the enzyme. So another way sort of showing that is, so the E1 component of this pyruvate dehydrogenase complex is tightly bound. All right, so that gets us.
Wait, Professor.
Again, there's three.
What happened to one of the carbons?
CO2.
Oh, thank you.
Yeah. So that's the so that is our first. That's our first decarboxylation, right? You know, in the whole glucose plus O2 goes to CO2 and water. This is the first time we now, in that whole process, we've actually generated CO2. All right, so now, again, there's three components, E1, E2, E3. So now we're going to get to E2. Which is also known as dihydrolipoyl transacetylase. Whew. So this also has, just like this E1 has a thiamine pyrophosphate is important, this one has its own kind of important prosthetic group, okay, which is lipoic acid, which is a sterified or an amide linkage to a lysine. So what does that thing look like? So we've got this is a redox carrier. It's going to carry electrons here. So this part of it here is known as lipoic acid. It's also nutritionally required, except that E. coli makes it so we can pretty much get it from E. coli. I don't think there are many examples of, I'd have to look to see if there are many examples of people who suffer from lipoic acid deficiency.
Professor, which part of it is the lipoic acid? The whole thing you
So
drew?
it's
Or?
so this is lipoic acid that is an amide bond to a lysine in there. So the lipoic acid would actually be the carboxylic acid here.
Okay, so it includes this part.
And so then this is a lysyl side chain. Remember lysine, that's the one that goes CH2, 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 NH3. Well, now it's in an amide linkage to this lipoic acid. And again, this ring is the business part, and it's basically because we can oxidize and reduce these sulfurs in that ring. And we're going to start with the oxidized version, and we are going to start with the oxidized version, and we're going to basically take those electrons from the thiamine pyrophosphate so I'm going to redraw the and now I'm going to put the business end of the lipoic acid attached to the E2. And what's going to end up happening, I'm not going to walk through all the mechanism in there, but basically what we're going to do is we're going to transfer this onto this sulfur group. And we're going to remake our so if we're following the this had come from pyruvate after the decarboxylation, and now it's being it's transmitted over or transferred over to that lipoic acid group. This is known as acetyl dihydrolipoamide. And again, all of this is ultimately connected to E2. So we've essentially transferred those carbons from E1, the thiamine pyrophosphate on E1, to this lipoic acid group on E2.
Professor, is that a G?
What's that? This
Is that a G?
here,
Yeah.
that's the negative charge for the
Oh, okay,
carbanion,
thanks.
sorry. All right, so the next thing that happens, so let's see, was E1, E2, the next thing is E3. And what E3 is gonna do is going to transfer this to another molecule called coenzyme A, and we're also going to transfer this to another molecule called coenzyme A, and then we're also going to reoxidize this to the oxidized form and transfer those electrons to NAD to make NAD extra high electron energy carrier. Okay, so 3, E3, which is dihydrolipoyl dehydrogenase. So the first part of it is relatively straightforward, is we're going to take our take that, and we're going to bring in coenzyme A. So here's another coenzyme, basically. Coenzyme, what the heck is coenzyme A? And why did I try to HS-CoA? Well, now I'm going to cheat a little bit, and I'm actually going to use a slide to show you the structure of coenzyme A. All right, good. Quick review here. There's our thymine pyrophosphate molecule that we showed the structure of. That's part of the E1. This was part of E2, the lipoic acid that connected to the E2 that is basically been the oxidizer force. Now we get into, we'll talk about that one a little bit, just a sec. Let me go ahead and get to, here we go, coenzyme A, coenzyme A. All right, so we start with a molecule that kind of looks like ATP. We have adenine, we have the ribose. It does have a phosphate on the 3' position. We go up, we have 2 phosphates here. Then it gets connected to this other molecule, pantothenic acid here, okay? That's another thing, I believe they put that in the Monster Energy drink. Yes, let's see. Yes, D-calcium pantothenate, vitamin B5. So this is another one, another vitamin here, pantothenic acid here. And then it gets attached to this beta-caproethanolamine, right? And then at the end of that is a sulfidyl group, an SH, right? So the way I deal with it in terms of drawing it is I just draw the SH and then CoA, just to prevent from like this. And then what happens, it basically serves as a carrier to make acetyl-CoA. Why is this important? Is this thioester right here is actually a relatively high energy bond. And so we can use that to drive the synthesis of some other reactions like in citrate synthase when we get into the citric acid cycle. So just recognize when I put CoA, I'm kind of cheating a little bit. There's a whole bunch of that molecule there that I'm not drawing out. All right. So we bring our coenzyme A here, and then that is going to give us our acetyl-CoA. Acetyl-coenzyme A. And then we have a fully reduced lipoic acid. So this would be a dihydrolipoic acid here. Again, this is still bonded to E2, but E3 is catalyzing this. So the next step is what we want to do is we want to take this in its reduced form. These are high energy electrons. What we're going to do is we're going to transfer those to ultimately to NAD to make NADH. Okay, so we've got that. We've got so the next step, though it's never simple, right? Is that there is another component of E3 that's diagrammatically represented like this. So this is flavin, adenine, dinucleotide. And then these are a couple of oxidized cysteines within that E3 protein. So this is all part of E3. So I'm going to kind of come down here to show. So we basically get oxidation here that's ultimately going to result in reduction here. Those electrons actually go through this FAD. I'm going to show you the structure of FAD in just a little bit. So so that we end up with the oxidized lipoic acid plus so this is the reduced form. Now we're not quite done, because now we have to take, again, we have some high energy electrons here in this reduced thiols. We have to transfer those electrons to NAD to make NADH. So the final step is I'm going to, let's see, I needed to plan ahead a little bit more. We're going to bring in NAD and make NADH. And that's going to regenerate our oxidized E3. Whew. So that gives our overall reaction that we end up with of this whole complex, E1, E2, E2, E3, is pyruvate to acetyl-CoA. We bring in coenzyme A, we give off CO2, and at the same time we take NAD and we make our high energy carrier NADH plus H plus. All right, so let's look at all the enzyme cofactors involved in this reaction, because that's sort of part of this story, is that E1, E2, E3, there's all these enzymes, and they end up having to use a lot of cofactors that ultimately have to be obtained from, in humans, we retain from our diet. So, cofactors in PDH. Let's see, we had the thiamine pyrophosphate. That was part of E1. What else? What was next? We had the lipoic acid. We have the coenzyme A. Part of E3. We have the FAD, and I'm going to show you the structure of that in a little bit. FAD, that also part of E3. Then we also had the NAD, which has the nicotinamide ring, which is a substrate for E3. Again, coenzyme A is where we need the pantothenic acid. Let me show you some of these other structures here. We'll just walk through them all. There's the thiamine pyrophosphate that we talked about, right? Then, let's see how many of these are the thiamine I don't see in here. There is, there's the lipoic acid. So this is part of the FAD molecule, and we're going to see this again when we get into the citric acid cycle, so I'll take a little bit of time. So this is another electron carrier. In this case, the electrons were going from the lipoic acid to those that part of E3 with the two oxidized or reduced cysteine residues. So basically what you have, it's a nucleotide again, base, sugar, phosphate. Then you have another sugar, ribitol here. And then the business end is this flavin here. So this whole part with the ribitol plus the flavin is riboflavin. So that's something else that's a nutritionally important molecule that we have to get in our diet. What makes FAD special, and I won't go through the details of this, except that basically it's a 2-electron reduction, so it can carry 2 high energy electrons, but it can carry 1 at a time. Whereas our other high energy electron carrier, our NADH, carries 2 electrons, but it has to take 2 at a time. So this takes 2 single steps. And so there are certain places where this molecule is more utility than the NADH molecule. And also the redox potential isn't quite as strong as the NADH. So the electrons that are being held in this molecule don't have quite the energy as NADH. So again, this is the nicotinamide. We've seen this molecule several times too. This is the NADH. We talked about this last time. This is this business end up here, this nicotinamide ring here that goes from the oxidized form to the reduced form. 2 electrons, that's with the high energy electron carrier. Again, this is the molecule that niacin, in our diet, we get niacin to be able to make this ring, or we can make it if we have enough tryptophan in our diet as well. Then here's the pantothenic acid and the coenzyme A and the pantothenic acid. So it's pretty cool. This reaction, which is right at the gateway to oxidative metabolism, requires all these enzymes and cofactors. Oh, so structurally, I'll finish up talking about PD. Well, no, because I'm going to talk about regulation a little bit. So this is a complex, and there isn't just one copy of E1, one copy of E2, and one copy of E3. There's actually multiple copies of each one, and they're arranged in a complex where right in the very middle is E2, and then more on the outside is E1 and then E3. So it basically, multiple copies come up in this sort of cubic structure, add together. So each of these complexes are going to have multiple catalytic sites basically going through there. And they're big enough that you can actually see them in electron microscopy. Most proteins aren't big enough that you can't see them with traditional transmission electron microscopy, but these things are big enough you can actually see them. Actually, I don't think I'm going to talk very much about the regulation of it. There are different regulatory mechanisms that can be down-regulated by phosphorylation. There's some feedback inhibition. It tends to be inhibited by NADH. That's one of them. And we're going to talk about the overall regulation of pyruvate dehydrogenase and the citric acid cycle after we get through the citric acid cycle. But I do want to take some more time now, having gone through this pyruvate dehydrogenase. Now we have this acetyl-CoA. We've made one CO2. We have to now further oxidize those other 2 carbons that are now attached to acetyl-CoA to CO2 and capture more of those high energy electrons in the form of NADH. So that's the citric acid cycle. So let me introduce the citric acid cycle. So first of all, let's just look at the overall reaction. We're going to take acetyl-CoA, and we're going to convert both those carbons into carbon dioxide, to 2 carbon dioxide. In the process, we are going to generate, take 2 NAD plus, make 2 NADH plus H plus. We're also going to take, actually, this is 3. We're also going to take FAD, and we're going to make FADH2, another high energy electron carrier. And we're going to take GDP plus PI and make GTP. So we're going to couple the oxidation of those 2 carbons to the transfer of high energy electrons to these high energy electron carriers, and we're going to synthesize 1 ATP from that. And we're going to generate the CO2. Now this is a cycle. So we're going to start with a molecule, oxaloacetate, and then we're going to regenerate it. So in our cycle, the first thing we're going to start with is oxaloacetate. Well, we'll start with our acetyl-CoA. We have our acetyl-CoA that we just generated from pyruvate dehydrogenase. And I'm going to draw the substrate, oxaloacetate, in kind of a funky way. This molecule here is called oxaloacetate.
That's the one we saw earlier.
That's right. This is actually the product of the pyruvate carboxylase reaction. Now we're going to essentially couple the hydrolysis of this thioester, which is a high energy bond, to the formation of another carbon-carbon bond. So we're going to end up and then what I'm going to do, just to kind of point, kind of like this, is I'll put a couple stars here to show you that, okay, these are our new molecules. We release the coenzyme A. This enzyme is citrate synthase. So that, and then this molecule here is citrate. Or if I draw it in the protonated form, it would be citric acid cycle. So that's how it got its name, the citric acid cycle. So now that we've done this, what the next of the reactions are going to do is we are going to basically end up going around remaking our oxaloacetate, but at the same time oxidizing and releasing a couple of these carbons as CO2. Okay, it's not necessarily those exact same 2 carbons that we added in there though, but there's a net loss of 2 carbons, and we remake oxaloacetate. So the next step is to take this citrate here, and we're gonna move this hydroxyl group down to this position right here. So it's just an isomerization. And this molecule is known as isocitrate. This enzyme is aconitase. It's named aconitase because there's an intermediate in this reaction involving dehydration here, a double bond called aconitate, but we won't get into that detail. But if you're just kind of curious about why the heck is it called aconitase, it's named for one of the intermediates in the reaction.
That's isositase.
Isocitrate. So this is citrate, now we just move the hydroxyl group over. Think of it as an isomer of citrate. That actually sets us up for an oxidative decarboxylation. So what does that mean? We are going to decarboxylate this. We're going to take this off and lose that as carbon dioxide. At the same time, we are going to oxidize that alcohol into a keto group, so that we end up going to